Hello, good morning. Can you hear?
Good morning, sir. Yes.
Hello. Hello, am I audible?
Yes, sir.
Yes, sir,
Oh,
you're audible.
okay, can you see the PPT?
Yes, sir. Yes.
Yes, sir.
Okay, that's fine. So today I'm going to start with the metabolism. You have already learned about the intermediary metabolism. You have learned whatever is the concept of anabolism and catabolism. And you know that how energy can be consumed and produced. So today I'm going to start with one of the simplest steps by which energy is being produced inside the cell. And here we are going to talk about glycolysis. And this glycolysis, you will learn, is very common, present from bacteria to higher organism. And it seems it is one of the most important energy producing steps inside the cell. Now, as name indicates, glycolysis. Glyco refers to the carbohydrate. Lysis means breakdown. So that means it is a catabolic process. And as you know, the catabolic process is the one which is going to produce energy inside the cell. So here, glycolysis is the sequence of reactions that metabolizes one molecule to glucose of glucose to two molecules of pyruvate. And what it does along with this is that concomitant net production of two molecules of ATP. That means when glycolysis occurs, glucose is converted into pyruvate and you get two molecules of ATP. I will give you whole reaction, all the set of events, whatever the way glucose is converted into pyruvate and production of two molecules of ATP. Now, if you recall, when we talked about the steps in intermediary metabolism, you have learned that most of our food items, they are consisting of a complex molecule, say, or you can say they are macromolecule. For example, carbohydrate exists as a starch or polysaccharides, other polysaccharides, whereas the proteins, they are also polymers. Nucleic acid, they are also polymers. So first step of metabolism is that you convert polymers into monomers. So that part, when you convert starch to glucose, that is the first step in the metabolism. And during this process, you do not get any energy. Second major step which we have discussed is that these small molecules, they are converted into a molecule which is further utilized for the energy production. So here the glycolysis comes at the second step. And in this step, you get energy, but not a lot of energy. You will learn that glycolysis prepares the molecule which is in the later stage of the metabolism, produces huge amount of energy. So if I'm saying that the end of glycolysis, you will be getting ATP, that means the ATP is not in large quantity. You get only two molecules of ATP. The major ATP is going to be produced from the pyruvate which you have produced, and this pyruvate will produce NADH and FADH2 as well as one molecule of GTP. Whereas during the steps of glycolysis, the energy production will be in the form of only two molecules of ATP, and each step is going to produce one molecule of NADH. So actually, the amount of the energy produced in glycolysis is not very large, but these are very quick steps. And you will be learning that there are many times when cell will compromise for speed of energy production over the amount of energy production. This part I will elaborate when we are talking about next few steps in glycolysis. Now you will, question why glucose has become a very important energy source. So you can see that the glucose in mammals is the only fuel which is utilized by the brain, and it is used under non-starvation condition, and is the only fuel for the red blood cells which can be used in the form of energy. So actually glucose is a very important source of energy. In fact, whenever you may have noticed that the brain does not get energy for even few seconds, you will feel, uncomfortable. You will feel, dizzy, and that's where the importance of glycolysis occurs. And that's why you see the patients suffering from diabetes who does not have capability to store glucose in their cell. Sometimes if they do not eat at proper time, they feel dizziness. So that is where the importance of glucose comes to play in a role. Now one of the monosaccharide formed from formaldehyde under prebiotic condition, this gives you a kind of a story how under prebiotic condition glucose might have been synthesized to become a source of fuel from primitive organism, and it has been retained as a source of energy up to the higher organism. Glucose has been chosen probably because it has a low tendency compared to other monosaccharide in terms of their ability to non-enzymatically glycosylate proteins. What does it mean? It means that normally you have learned that if you have a protein, it can form bond with the glucose molecule. So normally there are many such reactions which occurs where glycosylation reaction becomes important in the cell. Glycosylation means a carbohydrate unit binding with the protein molecule. Where does it bind? You can have N-glycosylation or you can have O-glycosylation. So N-glycosylation will occur wherever you have extra NH2 group. That means glutamate and aspartate. Where can you have O-glycosylation in the protein molecule? Wherever you have OH group. So that means serine, threonine, and tyrosine. So these are the residues where glycosylation can occur. But what happens? Glucose has a very low tendency. But when I say low tendency, it means it is not that they will not form bond with the protein molecule. Do you recall anywhere where glucose can interact with the protein? Any example? I can tell you a very interesting example, and that has been clinically also utilized for certain purpose. Normally, what happens if glucose level is quite high in blood, after a period of time, you will find the glucose forms a bond with hemoglobin, and hemoglobin gets acylated. So this glycosylation reaction where glucose binds with the hemoglobin is an important criteria to find out whether your blood level of glucose is high for a long time or not. And that is what is taken by the doctors to understand whether it is a 1 day fluctuation of your glucose concentration or for a long time. And that is known as HbAc analysis. So maybe some of you might have gone for such kind of analysis, but at least for those who are suffering from diabetes, in order to understand whether their blood glucose is higher for a longer time, they get HbAc analysis done. That is nothing but glycosylation of hemoglobin. But that happens when glucose concentration is present for a very long time, and it is high in the blood. Otherwise, the glycosylation level is very low. And that's one interesting point about the glucose. And glucose also has a tendency to exist in the ring formation, and that is why they have relatively less tendency to modify proteins. These are some interesting points about the glucose. Now comes another interesting thing which you will be learning, that the fermentation is a process which provides usable energy in the absence of oxygen. That means you recall when you are running, suppose you are running, what happens? your metabolism is much faster than your glucose can provide the energy. So under those conditions, what you find is that very fast reaction occurs. Your glucose is converted into pyruvate, 2 ATP is being provided, and though there is not a lot of energy, but some amount of energy which can be quickly supplied. I will discuss about the consequence. So during this process, what happens? This process is called as fermentation. So what is a fermentation? You may have heard in other courses or in some other places that there is a reaction fermentation. And very commonly fermentation has been associated with alcoholic fermentation or lactic acid fermentation. So when we run, what happens? Pyruvate is being produced. Pyruvate is converted into lactic acid, and those lactic acid are being accumulated until it becomes toxic. You can keep on producing energy. Otherwise, when oxygen is present, pyruvate is further converted, and finally it is converted into carbon dioxide and water. So that is a complete degradation of glucose molecule into their final products. So the final product of glycolysis is pyruvate, but the pyruvate can further undergo further oxidation process, which you will learn when we will talk about citric acid cycle or TCA cycle. Is it clear to you? So when excess of oxygen is present, the complete breakdown of glucose occurs. When oxygen is not sufficient, or in some cases reaction is happening or the conversion of glucose is happening in absence of oxygen, during that process fermentation occurs. So what actually fermentation means? So here I'm trying to explain you this process. So in the absence of oxygen, fermentation generates lesser amount of energy. Pyruvate is converted or fermented into lactic acid, which is known as a lactic acid fermentation, or into ethanol, which is known as a alcoholic fermentation. So in case of yeast, what you find that if I don't give a lot of oxygen, they will produce alcohol. So in yeast, pyruvate is further converted into your ethanol. Whereas in some of the lactic acid bacteria, you find that the pyruvate is converted into lactic acid. So they are called as a fermentation. I'll also define how we can define fermentation in terms of biochemistry. So fermentation yields only a fraction of energy available from the complete combustion of glucose. So that you have learned. You will be learning that at the end of glucose conversion completely into carbon dioxide and water. This is step. You get almost 36 or 38 ATP, depending on which mode of entry of certain molecules inside the cell is happening. So here, compare that in one process, this glucose converted into glycolysis, only 2 molecules of ATP is generated. Then pyruvate is converted into ethanol or into lactate. So these are the fermentative steps. And here, if oxygen is available, it can be converted into carbon dioxide and water. When animals perform, this is again explanation of the same thing. When animals or human beings, they perform burst intense exercise, their ATP needs to rise faster than the ability of the body to provide oxygen to the muscle. So you have less oxygen or limited oxygen. Under those conditions, what will happen? Muscle functions anaerobically. It means in the absence of oxygen until lactic acid builds up to the point at which fall in pH inhibits the anaerobic pathway. That means the ethanol, or so here lactic acid will accumulate till your pathway, this glycolytic pathway, is stopped or is inhibited. So it's a very interesting thing which you are learning here. First thing is that glucose. So in fermentation, what is happening? Glucose is converted into ethanol or glucose is converted into lactate. On the other hand, so what is conversion? One organic molecule is converted into another organic molecule, and it's an incomplete conversion. On the other hand, glucose is converted into CO2 and water, all the inorganic molecule. So here, what I can say here is, if I try to define fermentation in terms of a biochemist, it is incomplete conversion of glucose into another organic molecule. So one organic molecule is converted into another organic molecule, and it is incomplete conversion. On the other hand, when your carbon dioxide in water is formed during complete oxidation, that is known as aerobic metabolism, and that is where fermentation and oxidative phosphorylation can be differentiated. Okay, so glycolysis is basically an energy conversion pathway in many organisms, and what you see is that this pathway is common virtually in all the cell, in prokaryotic cell, in eukaryotic cell. Now question is, where does this occur? Where these reactions of glycolysis is happening? In eukaryotic cells, glycolysis happens in cytosol and comprising of these three steps or three major stages. So when glycolysis occurs, it converts. First step is that glucose is converted into fructose 1,6-bisphosphate. I will show you all the reactions here. Just I'm trying to tell you about the stages and their importance. So in the first step, the conversion of glucose into fructose 1,6-bisphosphate occurs. Here you will see that there is no energy being produced, rather the energy is being consumed. So you will see that ATP is being utilized in this step. You may question that this is energy generation pathway or this is energy generation steps. Why energy is being consumed? Answer is that during these steps, cell tries to make the molecules which in future can produce more energy rich molecules. So that's the way it is happening. And in this stage one, there are three sub-steps. One is phosphorylation, another is isomerization, and then again phosphorylation. So by these steps, phosphorylation, isomerization, and again phosphorylation, they are making fructose 1,6-bisphosphate. And here, as I said, glycolysis, these initial steps, they trap the glucose in the cell and form a compound that can readily be cleaved into phosphorylated 3-carbon units. So that's basically trapping the energy, glucose molecule. So first thing is that glucose should not move out of the cell. Glucose should be converting to the next step. So all those things we will talk in the next slide. Now, second stage is that the cleavage of fructose 1,6-bisphosphate into 2,3-carbon fragment. So here, 6-carbon compound is converted into 2,3-carbon compound. That means if I'm utilizing 2 ATP in the first step, and so I'm giving you a very simple example. Here in the stage 1, I'm starting with 1 glucose molecule. And if in each step, there are 2 steps where ATP is involved. So what is happening is that 2 ATP is being consumed. Now in stage 2 and stage 3, stage 2 is just interconversion. Stage 3 is ATP harvesting. Again, I am telling you that there are 2 steps where ATP is being generated. Now, the stage 2 is converting fructose 6-phosphate, 6-carbon molecule into 2 3-carbon fragments. So what will happen, even in those stage 1, 2 ATP consumed, stage 3, 2 ATP being produced, but at the end, what will happen? Can you tell me? In stage 1, a 6-carbon molecule is going to utilize 2 molecules of ATP. In stage 2, what is happening is that 1 6-carbon molecule is converted into 2 3-carbon compounds. And the last step, there are ATP harvesting. That means ATP is generated. And there are 2 steps where 2 steps where ATP is being produced. So stage 1, I'm saying 2 ATP being consumed at 2 steps. And stage 3, I'm saying there are 2 steps where ATP is being produced. Now you tell me what will be the net result.
So
you already know.
2 ATP will be produced in the end.
Yeah, because the number of molecules are doubled. And that is why in stage 3, there are total number is a 4 ATP because there are 2 cycles. And that's why you will get at the end 2 molecules of ATP being produced. This part you will learn slowly. But this is what gives you initial thought that how it can happen. Now I will be talking about individual steps. So what happens in step 1? Step 1, glucose enters inside the cell. And how glucose enters? It enters through the protein because glucose is hydrophilic molecule. It cannot enter inside the cell. So you will be having glucose transport proteins. That means you have a transporter, and as soon as glucose enters inside the cell, it is phosphorylated by ATP to form glucose 6 phosphate. In some of the bacteria, there is a very interesting reaction occurs where while entering inside the cell, this transporter protein itself works in such a way that the phosphorylation occurs in the membrane itself. So what I mean to say here is that glucose is designed to be converted into glucose 6 phosphate very quickly. Now question comes, what is the advantage of this reaction? How it is benefiting the metabolism of glucose? Number one, when glucose enters, unless it is converted into glucose 6 phosphate, because it is a small molecule, it has a probability of a diffusion across the membrane. And you can recall, being a very important molecule source of energy, glucose is being transported across the gradient. You know, it is not only along the gradient. Even if glucose concentration is higher inside the cell, they will be pumped inside the cell because it is source of energy. So what happens when glucose becomes negatively charged? So when you phosphorylate them, it becomes negatively charged. So inside the cell, what is the potential? Inside the cell, you have a negative potential. And that is why your glucose molecule will not diffuse out. They will be repelled inside the cell. So they will be forced to remain in the cell. So that is one very important point. Second thing is that when you add phosphoryl group, that begins to destabilize glucose, and thus it helps in the further metabolism. Though I have not written this point, this phosphorylation reaction here that also helps in binding with the enzyme, because glucose phosphate, once you make glucose phosphate, it becomes negatively charged. And then it can react with the enzymes where the groups or the active site has a positive charge. So they will start interacting with the enzyme. So these things are very important for the interaction of glucose with the enzyme, with the within the cell, and that's where they are being retained. Now, next step is that the kinases. Kinases is a general name for a group of enzyme. They can transfer phosphoryl group from ATP to an acceptor molecule. So as I said, that the first step is going to utilize ATP, is going to consume ATP. So what you see here is that ATP will transfer its phosphoryl group to glucose molecule. So it is basically a transferase reaction where phosphoryl group is being transferred. And you will see that the hexokinase, this enzyme, that always requires magnesium ion as a cofactor. So magnesium in some cases, they also require Mn+2, and that is what is required to these kinases, they are required for the activity of enzyme. As you know that many coenzymes or cofactors are required by the enzyme. So here, magnesium ion is required. This basically helps in the binding of ATP with the enzyme and then transferring its phosphoryl group to glucose molecule. So it's an interesting reaction. Now, some of you may have a question, why we call them as a hexokinase. Number one, that it reacts with its substrate is a hexose. Now, for your information, though I have not written here, this enzyme is a general enzyme which can metabolize glucose, which can metabolize mannose, which can metabolize galactose. It can also react with fructose. So it is a common enzyme which reacts with many sugar molecule, 6-carbon compound. The only thing is that all of them should be 6-carbon compound having, carbohydrate property, where hexokinase can transfer its phosphoryl group to glucose. Now, there may be a difference, and that maybe we can discuss later on. the binding of glucose to hexokinase, how glucose binds with hexokinase, that will decide that how, how much reaction of one of the substrate can move on. To explain it better, let me tell you. Suppose I am carrying a reaction where I have a glucose, I have a fructose, and I have a mannose or maybe galactose. All of them, if they are present together, and I put hexokinase as an enzyme, what will happen? Glucose will be metabolized first. Can you tell me why? Or to give you a hint, glucose will bind with hexokinase at low concentration. What does it mean? Can you recall something? We have discussed this point. Okay, another hint is that the binding of a substrate to enzyme gives you value of Km. Do you recall Km?
Yes, sir.
Km is the binding affinity of the substrate with the enzyme. So what I mean to say here is that the Km of glucose, fructose, mannose, and galactose will be different. Now you tell me, after telling you all the story, that which one will be having the lowest Km value and why? The one which is having the highest affinity will have lowest Km value. That means glucose will have lowest Km value. So even if I have glucose, mannose, galactose, etc., the glucose will be utilized first because it is having lower Km value. Am I clear?
Yes, sir.
Okay, let us move on. So once you have learned this story, let us talk about how the formation of glucose 1,6-bisphosphate occurs from glucose 6-phosphate. So you have seen that glucose has been converted into glucose 6-phosphate utilizing hexokinase. Though there are other kinases, you will be learning that there is a specific kinase about the glucose kinase. You can have fructose kinase, but this hexokinase is a common kinase having different Km value for different substrates. Now, coming to the next step where fructose 1,6-bisphosphate uh, has to be formed, that is, if you look at fructose 1,6-bisphosphate, it is having two phosphate residue, and glucose phosphate has one residue. That means it has to again, you have to have an enzyme which can again transfer one phosphoryl group. So we are going to talk about that. So let us see how it happens. And another thing which you are learning here is that glucose 6-phosphate is converted into fructose 6-phosphate. Glucose and fructose, they are isomer. And what kind of they are isomer? They are aldose ketose pair. So let us see what happens. The next step in glycolysis is isomerization of glucose 6-phosphate to fructose 6-phosphate. So first step is conversion of glucose 6-phosphate into fructose 6-phosphate. Both of them, they are isomer. The only difference is one is a aldose containing molecule, another is a keto group containing molecule. And when such conversion occurs, the mechanism is that glucose 6-phosphate first converts into open chain, and this open chain interconversion occurs, and then again they will go under closed chain. And here what you see is that instead of the ring structure that changes between glucose and fructose, it is because of the aldo keto pair. So the isomerization of glucose 6-phosphate to fructose 6-phosphate is conversion of aldose into ketose. The enzyme phosphoglucose isomerase. So here enzyme is isomerase enzyme, and it is called as a phosphoglucoisomerase because glucose is phosphorylated here. So you have seen the conversion of glucose 6-phosphate into fructose 6-phosphate, and as an intermediate step, you will find that the 6-member ring of glucose 6-phosphate is opened up before it is and then isomerized, and after isomerization, second phosphorylation reaction occurs. And what happens that fructose 6-phosphate is phosphorylated by ATP to fructose 1,6-bisphosphate. It's a simple reaction. So you can see on the top, glucose 6-phosphate has been converted into fructose 6-phosphate. And fructose 6-phosphate in presence of phosphofructokinase, again requiring the similar kind of a cofactor, and ATP will transfer its phosphoryl group, and then you have one phosphoryl group additional in fructose 6-bisphosphate. So in the first stage, you have seen that there are two steps. First is conversion of a glucose to glucose 6-phosphate, where ATP was involved, and second is conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Conversion of this reaction, glucose 6-phosphate to fructose 6-phosphate, does not require any ATP or energy. Let us see the next step. Next step is a very simple reaction, but it is very interesting. What you are doing is that you are converting 6-carbon molecule into 2 3-carbon compounds. So here I have given you a reaction which is being catalyzed by the enzyme aldolase. Why it is known as aldolase? It is based on the reverse reaction. What you are seeing is that here you have a compound which is 6-carbon compound, and here it is making 2 3-carbon compound. And what you have in these 3-carbon compound, one is aldose, another is a ketose. So whenever this one aldose and one ketose reacts and condenses, that reaction is known as aldol condensation. You can recall in 12th class you have been taught about the aldol condensation. So reverse reaction is aldol condensation, and that is why probably the name, trivial name of enzyme is aldolase. So what it does is that it breaks the molecule from the center. So 3-carbon on one side, 3-carbon on the other side. And at the end, what you get is that you get one aldehydic molecule, which is a glyceraldehyde 3-phosphate, and one molecule which is having keto group. So it is a dihydroxyacetone phosphate. Now you look at these two reactions. Both are 3-carbon unit. Both are isomers, and both of them, they can be, both of them you find that if I look at the number of carbon, number of phosphate group, everything is same. So they are basically isomers. So what kind of isomers they are? They are aldo-keto pair. That means if I have an enzyme, they can convert them between the dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So actually, if you look at, if I take this reaction and carry out in a flask or in a test tube, what you will see is that this reaction, which is catalyzed by the aldolase, is a reversible reaction under intracellular condition. So if I give all the intracellular condition, this reaction is reversible. So you must have a question that why fructose 6-1,6-bisphosphate is metabolized, why it is moving forward. So ultimately what you will learn is that the reaction beyond this, after formation of glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate is utilized for next step of the reaction that we will discuss. But important point is that glyceraldehyde 3-phosphate is removed from this equilibrium reaction. So can you recall your Leach-Atelier's principle? Where if I remove the product from the reaction will move in the forward direction, and that is what is happening. Very interestingly, these two interconversion occurs in presence of isomerase enzyme. Because it's a keto, it is aldehyde. So what will happen, because glyceraldehyde is removed from the reaction, all of the dihydroxyacetone phosphate will be converted into the aldehydic form, and that's the way your reaction moves in the forward direction. So I have shown you how this conversion occurs, because both of them are triose phosphate. Triose means 3-carbon containing molecule, and phosphate. So the enzyme involved here is a triose phosphate isomerase. And I will tell you some very interesting point, that this reaction in presence of enzyme is very, very fast reaction. And that is why I'll just show you a glimpse of how the mechanism occurs. What is the basic mechanism? Why you require that enzyme? And how this enzyme helps the metabolic process. So triose phosphate isomerase salvages a 3-carbon fragment. So you have a glycerol 3-phosphate, which is present in the glycolytic pathway. As I said, that they are the one which are being utilized in the further reaction, whereas a dihydroxyacetone phosphate is not. So what happens, this dihydroxyacetone phosphate will keep on converting into glyceraldehyde 3-phosphate. And the glyceraldehyde 3-phosphate will be utilized in the future reactions. These compounds are isomers and they are rarely interconverted, but you know that their reaction, interconversion is a reversible reaction. The hydroxyacetone phosphate is a ketose, whereas the glyceraldehyde 3-phosphate is an aldose. We have already talked about. Now we look at the isomerization of 3-carbon phosphorylated sugar, which is catalyzed by the enzyme triose phosphate isomerase. I'm writing it as a TIM. This triose phosphate isomerase catalyzes the transfer of hydrogen atom from carbon 1 to carbon 2, and I will show it to you how it happens, to form dihydroxyacetone phosphate to glyceraldehyde 3-phosphate by intramolecular oxidation reduction reaction. This is very interesting. I'll show you in the how it happens. Now, isomerization of a ketose into aldose proceeds through the enediol intermediate. So what happens? This enediol is formed as intermediate. Now, this enzyme also does a very important job, that it does not allow non-enzymatic removal of a phosphate. Normally, what can happen, that this enediol intermediate can remove its phosphorus and it can form methyl glyoxal. And if methyl glyoxal is formed, all your reaction will stop, because you will not get enough of glyceraldehyde 3-phosphate, and that's why you cannot proceed in your metabolism. So this step becomes very, very important. I'll show you how fast this reaction is in presence of enzyme. So there are two features of this enzyme, TIM, that is triose phosphate isomerase. One of them is that it displays great catalytic process. But what it means is that its power of catalysis is very fast. It accelerates the isomerization by a factor of 10 to about the 10. Now, how we compare? We compare from the non-enzymatic reaction. So you will see that most of the time we talk about the reaction, and the reaction rate may be very, very slow. But let's compare it from the rate which is obtained by a simple chemical catalysis. So if I look at simple base catalyzed, such as acetate ion, which catalyzes this reaction, and the TIM presence, that means in presence of enzyme, you will find the rate of reaction is 10 to power the 10. That means absolutely the reaction which is not happening can be catalyzed at a very fast rate. And its Kt by Km has been calculated. What is Kt by Km? Kcat by Km? What is Kcat by Km? What is the significance of Kcat by Km? It gives you catalytic efficiency of enzyme. That means what you require very high Kcat value, the value which shows you how your product is formed, and very low Km value, that means how fast it can interact with the enzyme. So your substrate binding with the enzyme and the release of a product from the enzyme, these two gives you catalytic efficiency. We have talked about this in the class of enzyme. And what you is that the ratio of isomerization of glyceraldehyde 3-phosphate is something like 2 into 10 to the power 8 molar per second. So you will find that 2 into 10 to the power 8 molar per second is the value of Kcat by Km, which is a close to diffusion control limit. That means beyond this, you cannot increase the rate of reaction. Then only you can increase the rate of reaction by increasing the diffusion. That means interaction. You force the interaction of the substrate with the enzyme. So these are all those things which we have already discussed in class of enzymes. So here we are going to talk about the another advantage of TIM. TIM suppresses an undesired reaction, that is decomposition of any diol intermediate into methyl glyoxal and inorganic phosphate, as we discussed over here. So here any diol can be converted into methyl glyoxal, but if it is present, this intermediate is bound on the surface of the enzyme, it will not allow them to remove PI non-enzymatically. Rather, this PI will remain bound and they will be utilized for the formation of the product. Look at these reactions. These reactions are not, I'm not expecting you to learn enzyme kinetics at this point, not the enzymatic mechanism, but it will show you, which we discussed in the class of enzyme. I purposefully skipped giving you the mechanism, but what you see here, this is the enzyme surface where the reaction is occurring. So you have enzyme. To this enzyme, your substrate is binding. So dihydroxyacetone phosphate is going to bind. Now it has to be converted into glyceraldehyde 3-phosphate. How it happens? This dihydroxyacetone phosphate, once it binds, the reaction starts. The glutamate 165. What does this 165 means? 165 is the position in the structure of a enzyme where glutamate is present. So 165, 195, it indicates the presence of the amino acid in a particular enzyme. So what happens? Glutamate, it is having, it will give its proton and then the reaction initiates. And what happens? Ultimately, this is taken up by the histidine 95. Histidine 95 is the one which is at the 95th position. Your histidine is present. Now, at this point, I'll give you a question and I would like to answer from you. Histidine is present in many, many enzymatic active sites. What could be the advantage of presence of histidine? The clue for you is that histidine has got its, functional group has pKa value, which is 6.5. What does it mean? When 6.5, it is very close to pH 7, isn't it? That means histidine has a property where it can either act as an acid or it can act as a base. So you will find that the many enzymes, they will have histidine at their active site. So these are the questions which I'm trying to answer you during the process of real reaction which is happening inside the cell. So inside the cell, what you have is that you have pH 7, where you have histidine. It can act as an acid or it can act as a base. So what you will find is that during this reaction, the dihydroxyacetone phosphate is isomerized and converted into glycerol 3-phosphate. So that's the set of the reaction. You will be learning about the enzymatic mechanism at the later stage, but this gives you some very basic understanding how everything is happening. Another very interesting concept which you should learn, that because it is having these reactions are occurring inside the crevices or cleavages present in the active site, that is why they will avoid presence of water, and that's why the interaction can be much, much better than it can happen otherwise in presence of water medium. So the triose phosphate reaction is the second step where it can interconvert your substrate molecule in such a way that you have excess of glyceraldehyde 3-phosphate. Because glyceraldehyde 3-phosphate will be further utilized, and that's why dihydroxyacetone will, so the reaction will proceed towards the formation of glyceraldehyde 3-phosphate. Now, the last step for the energy generation will be the formation of your glyceraldehyde 3-phosphate will be converted, it will be phosphorylated, and the step is known as oxidative phosphorylation. I'll be talking about this third stage in the next class. If you have any question, please ask. In this step, where energy transformation occurs, so these are important steps. These are the steps where actually energy will be generated or energy forming molecules. So you get NADH in this reaction, which will further produce ATP in the oxidative phosphorylation. That part we'll discuss separately. But what you get as a resource is NADH and ATP. So we'll talk about that. Anything you have, any question we can talk about. So what we have done is that we have talked about stage 1, where glucose can be converted into glucose 1,6 bisphosphate. You have seen one molecule of ATP. utilized. Where does this 1 ATP is being utilized? In formation of glucose to glucose 6-phosphate. And second molecule of ATP has been utilized for conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. We have also talked about stage 2, where fructose 1,6-bisphosphate has been converted into glyceraldehyde 3-phosphate as well as dihydroxyacetone phosphate. We have also talked about the interconversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate will further enter into another stage where actually you will see the production of NADH and ATP. So we'll be talking about these reaction, and you will learn how this reaction is important. Now I'm giving you at the end a question, and the question is that if I'm just going back to the reaction, and this question is for you to think over. Yeah, the stage. Yeah, in this stage, suppose suppose there is no oxygen, and also there is no production of ethanol or lactate. What will happen to glycolysis? Or in other words, if I ask you a question, is this reaction where glucose is converted into ethanol or glucose converted into lactate? These lactate and ethanol are end product of metabolism, or they have certain purpose to solve in these reactions. So this is a very interesting point we will discuss at the end when glycolysis chapter is over. Okay, any question? Any one of you? So if there are no more questions, I would like to thank you, and we will continue with glycolysis in the next class. We will also discuss some of the integrities, some of the interesting point about the glycolysis. Thank you very much. I will sign off.
Thank you, sir.
Thank you, sir.